Okay, so I want to go through a little bit of why the genes might be under transcription control the way that they are. And for this, I want you to think about the functions of the genes involved. So let's go over to an example here. All right, so let's say we've got, I'll keep those same symbols as before, circles for the substrate, diamonds for the product. If a cell finds itself with a food that could be digested to generate energy, then it helps to have the food, the presence of the food, be the signal that turns on transcription. Try to wing it over here. So food being present causes a change in this regulatory molecule. Maybe think about what kind I've drawn here. So it attaches to this molecule, and that caused the molecule to enable transcription. Oops, put my interacting molecule in there. What this means is that by attaching to the DNA, it turns transcription on. That would make it, by definition, an activator molecule. So now we have an active activator that's giving us plus, 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 net plus, turns on transcription of a, I'll draw a little protein here, digestive enzyme. And that would cause this process to happen here where the food molecules are digested and turned into energy. And that same system, under all the same controls, the food ran out. So let's say the digestive enzyme came in and successfully went through and digested all of the food particles, or at least most of them, turned them into the product molecules. If the food molecules were in low abundance, try to replicate our diagram here, digested. Let's do just no food. If the food molecules are gone again, or if the food molecules were never there, then nobody's there to interact with the activator. So the circle molecule that was interacting with the activator before to activate it is not there, then that keeps the activator off and prevents it from attaching to DNA. And transcription is off, and no enzyme is made to digest. No enzyme is made to digest the food that doesn't exist. So the two states of this activator, being activated on the left or inactivated on the right, really control whether the molecule is made, the enzyme is made that would digest the food. Yep. Okay, so we have an inactive activator, a minus plus there. So what I wanted to make me illustrate by drawing this was that the food situation of digesting something for energy is better controlled by the presence of the food itself. So the food is the signal that it should be digested. And this is the system we call inducible. So usually off, not just going around all the time preparing to digest food that's rarely encountered, but just turning on the enzyme digestion when needed. So inducible, this is the process of digesting, basically. or breaking down molecules. The other situation of repressible that's a different situation where the process of making something use a fancy term there. So there might be a biological process of making something that is needed. So the examples we're going to use involve an amino acid. So if a cell is making proteins, it's going to need amino acids as the building blocks for making those proteins. But as a manufacturing process, the manufacturing process needs to make amino acids only as much as they are needed. So once amino acids are made, they go off and become proteins. And if this process slows down, let's say the cell isn't doing a lot of growing, maybe it's going into a dormancy, then it's not useful to keep making amino acids if they're not always being used. So having a little bit of amino acid present maybe is a signal that there needs to be more. And having a lot of this present means that it's built up to an amount that there's more than the cell needs. So I'll split myself into two here. Let me just get us some more space down to the bottom here. So substrate becomes product. And this question of whether this product is being used. So rather than breaking down a molecule for energy, which we saw before, we're building up a molecule for use in the cell. So if there's not a lot of that, there's not a lot of the product, then that's a situation where there should be transcription. So I'm going to stick with the example of an activator. If there's not a lot of product, then the activator attaches to the DNA of the promoter for the gene that makes the enzyme. And this is the enzyme responsible for converting circle into diamond. So this would make more diamond. If we had a situation where there was a lot, then the diamond interacts with the activator and prevents it from activating. So no enzyme, and therefore no new product. So we have a switch again. If there's not a lot of product, the signal basically happens to make more product. If there is a lot of product, then the signal happens to stop making more product. So it's also a system where the machinery that turns on transcription responds to what the cell needs, and in this case can turn it off if there's not a need for that enzyme. All right, this is the kind of thing you probably will need to work through on your own just to make it make sense for yourself. So we've seen inducible expression. This is usually, or in our simplified examples at least, this is going to be dealing with molecules that can be broken down for energy. And then repressible gene expression. These will be molecules that are being built up for use in biological process, amino acid or nucleotides or something like that. And having too many of them becomes the signal to turn off transcription. And then here's our little repression example. This is what an enzyme looks like when it is being repressed. All right, so let's work through some of these here. Ideally, you could maybe answer them on your own before I tell you the right answers. So showing a diagram here, is this a gene being induced or repressed? So basically going from the top to the bottom, it's off in the absence of that molecule, and on the bottom, it's then on. And so that is a situation where the gene expression is being induced. So it's turning on, it's normally off, and then it turns on. So it's sort of being turned on. Induced is one of those positive words. So expression is being induced in the presence of the purple circle. The green molecule, I try to throw you once in a while one that isn't color-coded, but this one is color-coded, so the green molecule is an activator. Even if it wasn't color-coded, what you would see to know that it's an activator is that when it attaches to DNA, it enables transcription. That's really in the definition of being an activator. And then finally, the gene is induced in the presence of the purple molecule. And so for my shorthand there, the purple molecule is the substrate in this reaction. So substrate product, substrate of the reaction is interacting with the activator. Okay, so let's get some specific examples. It's kind of a really classic example that I'd say just about every biology class encounters when they talk about this topic. It involves the digestion of lactose. So lactose is a sugar, a disaccharide, and it's not one that bacterial cells commonly encounter. So again, it's one of those that they kind of need to turn on digestion of under certain circumstances. So if they find themselves needing lactose and they have lactose around them, then they can turn on the enzymes to digest it. Otherwise, most of their life, even if they're encountering sugars in their environment, they might not be lactose, and they might not need the enzymes required for that. It's a little bit, there's another layer of complexity to this, which involves having the enzymes, not just the enzyme for digesting lactose, which is this one called beta-galactosidase. There's also an enzyme for getting the lactose into the cell. So permease is an enzyme that we'll see is involved in this process, and beta-galactosidase. And honestly, I think I probably just have it that we don't think about this other molecule, allolactose. There's a little bit of a conformational change in that, but we don't have to worry about that. So big picture, lactose gets digested in the presence of beta-galactosidase. That's its job. Okay, once digestion happens, then we have these two monosaccharides, simpler sugars, galactose and glucose, and those are easily digested by some more mainstream enzymes that might well be on all the time. Okay, so as I said, permease and beta-galactosidase. So if these are enzymes, enzymes are proteins, and proteins have genes that are the instructions for making them. So there'd be a gene for permease and a gene for beta-galactosidase. And it turns out, something I don't think we've really seen before, that bacteria have a neat structure that they can make called an operon, where they grouped together genes that have similar related functions, and they can all be transcribed together. So this harkens back to a term we encountered a while back, polycistronic. Those were those gene regions where there's one promoter that causes transcription of more than one gene. And so we have this promoter located here. There's one promoter, and as a result of transcribing from that promoter, the LAC-Z, LAC-Y, and LAC-A genes are all transcribed on the same RNA, and then independently transcribed into same colors. So LAC-Z becomes beta-galactosidase, LAC-Y becomes permease, and LAC-A becomes transacetylase. And all of those together are involved in the process of breaking lactose down into glucose and galactose. So don't get too bogged down in the details of LAC-Z, LAC-Y, LAC-A. I'm not going to ask you to memorize those terms. They're they're really just arbitrary names. So LAC-Z, LAC-O, LAC-I, we're going to see a bunch of those. You don't have to know them from each other. I'll tell you what they do. I'd rather have you focus on what the big picture is and the functions of the process of gene expression a little bit more than the fine details of digesting lactose. Okay. Whew. All right, so as I mentioned here, LAC-A, LAC-Y, LAC-Z. Turns out this transacetylase, at least from what I've looked up, couldn't find out what it actually does. Kind of interesting, but we talked about the permease and the beta-galactosidase already. All of them are involved in lactose digestion. All right, we kind of talked about this, but just in case you were fast forwarding during that portion, if this then is a polycistronic operon, one promoter for each gene, well, you can just judge by the illustration here. So this is one promoter, and that one promoter covers all 3 genes. So each gene doesn't have its own promoter. Promoter, again, involved in transcription. Translation would happen independently. So LAC-Z would be translated separately from LAC-Y, from LAC-A, and that would involve the ribosome zipping through here at in a different process than the ribosome going through here and then going through here. So that was false, this question. All right, inducible or repressible. This is tricky because I haven't really diagrammed it right for you. Maybe think about that a little bit later. Let's, I don't think I gave you the right diagram for this one. Inducible or repressible gene expression. Let's hold on to that thought and check the next